Hello, today we're going to go over the answers to yesterday's assignment, which was HeLa cells, and looking at our aspects of cancer that we looked at before at the beginning of the unit and applying them to HeLa cells specifically. And you should have watched a video from yesterday, what HeLa cells are, but just a quick review from that. We have HeLa cells, which are taken from a woman in the 1960s who had cervical cancer caused by HPV. So these are cervical cancer cells that laboratory people who worked in labs, researchers actually figured out these could just kept replicating and replicating and replicating, making them great study systems because they replicated so quickly and they were able to give them to labs readily. They never stop dividing, which is a problem for other types of cells that we use for cell culture. They can stop dividing after normal 50 divisions, which is what this first excerpt gets into. So we have excerpt 1. For aspect of cancer, this would be, and there's more than one answer to some of these. Some of these can be interpreted to where you could find multiple aspects of cancer. And that's true of most science, that you might see more than one concept coming into play here, which is totally fine. So if you didn't have this one specifically, just add it on there and keep the answer that you already had, because it doesn't mean it's wrong. So this aspect of cancer in excerpt 1 was talking about telomeres and the fact that cancer cells never stop dividing, especially HeLa cells. HeLa cells never stop dividing. Whereas our normal cells, so normal cell behavior was about 40 to 50 times was their max replications they could do, and then they died. Whereas these guys just kept going and going and going. And the evidence for that was that they're still being used in labs around the world, and they still haven't stopped dividing. Next, our excerpt 2 aspect of cancer, we had a mutated p53, which is our tumor suppressor gene here. And when you have a mutated p53, remember those are like the guardians of the genome is what they call them in multiple of these videos we've looked at, because they're checking for mutations. And if they find mutations, normally, if you have a working p53, they find mutations, they send that mutated cell to be repaired if the genome can be, that mutation can be repaired. Or if that doesn't work, then send to apoptosis. Okay, so that's how normal p53 works, is usually find it, check it, nope, mutated, can't fix it, send it to apoptosis. But HeLa cells have a mutated p53, and HPV was a direct cause of that mutation of p53. So that human papillomavirus interfered with the p53, making it non-functional. So that's our aspect of cancer, is mutated p53. And specifically, that would be our evidence, is the HPV, human papillomavirus, shut down that p53 gene. And that allowed mutations to accumulate in her chromosomes, making her have those 76 to 80 heavily mutated chromosomes. These mutations just keep building and building. And the more HeLa cells divide, the more mutations accumulate. Normal cells have 46. She has almost double that, which is crazy. I mean, that's a lot of mutations and extra DNA, somehow a lot of mutations there. And we're going to look at her karyotype with looking at those chromosomes later in this video. Excerpt 3, this was talking about fast replication and reproductive rate. So if you had that, that's probably what a lot of you had because of the first sentence. However, it was talking more specifically about this lack of apoptosis. So if you have, in normal cells, you have telomeres shortening and cells eventually going to apoptosis once those telomeres are short enough. Remember, once those telomeres are short enough, they're getting into the good stuff of the DNA. We're actually getting into stuff that makes proteins we need. Therefore, for normal cells, this will be sent right to apoptosis. But that's not what happens in HeLa cells. She has that telomerase enzyme that just keeps adding to the ends of the chromosomes. And when it's adding to the ends of the chromosomes, you actually get basically it's what they call in this excerpt the elixir of life. They're not dying, they're not being sent to apoptosis because the aging of the chromosome is not happening. The telomerase would be your evidence. Telomerase adds to the ends of each chromosome. It's not sent to apoptosis. And her cells are older than Henrietta was when she died, which is crazy. Excerpt number 4. On excerpt 4, we're talking about, we have this HeLa line, and this high rate of reproduction and a lack of contact inhibition. So that's the aspect of cancer we're looking at. You could have high rate of replication, reproduction, or lack of contact inhibition. Both of these were being talked about here. Normal cells stop dividing once they contact each other. But HeLa cells, not only do they reproduce extremely fast, that they take over other cells in terms of the rate, but also if they touch another cell, they don't stop dividing. They just keep dividing. But normal cells, once they touch another cell, they stop dividing. So HeLa cells don't have contact inhibition and do have a high rate of reproduction. The evidence for that was that they take over other cells in human cell culture. Okay, now what you're doing is you're basically identifying, we took each of these different excerpts and matched them to a different aspect of cancer. Now we're going to match these excerpts best to the diagram they're describing. So if you had to have a diagram that described each excerpt, which one would you choose? And I'm not going to go into the whole evidence and reasoning thing for these, but I'm going to tell you which ones match to which. And just like before, there can be more than one answer for these. So I'm going to give you the answers that you could get for each one. So we're going to, number 1, this first one, this aspect, and you'll see all these diagrams that we're going to talk about we've seen before. So we're just matching them to the HeLa cell aspects today. This one connects to excerpt number 3. So cells avoiding apoptosis in cancer cells, you have those mutations building up, and they're not being sent to apoptosis like it shows for normal cell division. They're being sent to apoptosis. Cancer cells are not going to apoptosis once they're damaged. They just keep replicating and replicating. So that would best fit with excerpt number 3. For the next one, sorry, let me scroll down. This one would best fit with number 2 because we have mutations accumulating with each division. And the way we're able to get mutations accumulating is through that P53. That P53 is enabling more and more mutations, and that's what you're seeing here. So second mutation still allowed to be around because of mutated P53. Third mutation, so we're getting a little bit wonkier and wonkier. Fourth, and then eventually we have these uncontrolled, non-functional, mutated cells, and that was allowed to happen because of P53. So this one would best fit with number 2. You could fit multiple to this one again, but this one fit best with number 2 just because it shows how the mutations are accumulating. All right, next one we have this picture. This one would best fit with number 2 or 4 because yes, we have P53 happening again, and it shows in here a mutated tumor suppressor gene, which allows the copies that are mutated, but it's also showing this clump of cells, which is showing a lack of contact inhibition. So this one would also be number 4. I saw number 2 or 4 because this mutated tumor suppressor gene and also this clear lack of contact inhibition here, where it shows normal cells just even line once it doesn't enable them to clump up because they stop dividing once they touch each other. Whereas this is clearly clumped up and it did not stop dividing once it touched another cell. So that would be number 2 or 4. This last one we've seen before, and I know your paper it was a little bit blurry on there, but we have total cell counts. Remember, this is just like a total number of cells in a container So think of this as just a number, like you could count 1, 2, 3. I mean, there's a lot of cells per container. And then we have cells per milliliter. And we talked about this graph. We talked about it as a little, like, think of it as a test tube, and it's full of cells. So a test tube full of cells. And the more you go to the right, the more cells you have in this test tube. So this is a way of showing density dependence. So this one would fit with number 1 or 4 because it's showing a really, for cancerous cells, a rapid rate of reproduction. They're going much higher and faster than normal cells. However, it's really showing contact inhibition and a lack of contact inhibition because normal cells, once they reach this high density of cells per milliliter, their reproduction goes down. But cancer cells, you can see, they don't care. So if they just keep multiplying in this test tube over and over and over again, and even if they start touching each other, they just keep multiplying. So they don't care if they're touching another cell or not, which is why you see this really exponential rate of increase. So number 1 or 4 would fit that best. Okay, last thing is HeLa's chromosomes. So you can see, ooh, pretty colors. This is just a diagram that shows a normal karyotype on this side, and then what HeLa cell karyotypes look like. So remember, a karyotype is a picture of chromosomes. So we're comparing a normal cell's chromosomes to HeLa cell chromosomes. And similarities, what you likely saw was that they have a general same shape. You know, they're generally shaped the same. They have that, they get skinny. In the center, we call that the centromere. Remember when we were mapping karyotypes earlier in the semester? These guys are thicker and they're they still have the skinnier part. They still have that centromere that they can bend by. And remember, the centromere is how they bend over. So we have the same general shape, the same number of chromosome types. So you can still see there's 22 types of chromosomes in both pictures. But clearly there are a lot of differences here. So there's a lot of things you could put here, but probably what some of you guys put is there's a difference in the number of chromosomes for each type. For example, number 1, in a normal karyotype, you're supposed to only have 2. Remember, 1 from mom, 1 from dad. However, in HeLa cells, she has in these cells tons of chromosomes for each type. So 5 types for chromosome 1. That's really wonky. That's caused by a lot of accumulated mutations. In no way is that supposed to happen. And that's true for most of these. She has at least 3 or more copies of each chromosome of each type. So that was a big one that you probably noticed. They're thicker. So that shows the different mutations that have occurred is the thickness of the chromosomes. They don't have that, they don't necessarily have the same density or thinness. They are a little fluffy. They appear fluffier. And that was probably the biggest thing, is that you have, not everyone looks identical of each of the different types. There's more than 2 of each type, and they were like thicker and clearly more mutated. Got that fuzzier shape here. This aspect of cancer would definitely apply to the P53 gene and the mutation there, because the only way you got all these mutations was because of the P53 that was mutated, so it wasn't guarding the genome for mutations anymore. But it's also the telomerase. Remember that the telomeres added to the end of the chromosomes. And that's probably how we're getting so much DNA in each of these chromosomes, is that enzyme that keeps adding to the ends of them. So that would be the end of this one. This was a general review of the aspects of cancer, and we're just applying it to a new context with HeLa cells. There's a lot of videos you go watch about HeLa cells if you're curious about it. That's all we have time for in this section. But if you're curious, I can refer you to a lot of books or videos, or there's a couple movies. There's a movie on HBO about this too. It's a cool story if you want to look into it. I'm also going to post another video about meiosis. So go ahead and watch that today too. Okay. Bye.